Okay, I can't see myself. Broadcast is live. Why isn't my camera on?
Supposedly live broadcasting, but I can't see myself. All I got is this is fun. Technology.
I mean, there's no connected camera.
What? This computer doesn't have emoji, but it has those guys. Whatever that is.
What the hell is that?
Dang it.
Oh, that's almost better.
Yeah, it's even
That's like
weirder.
old school emoji. That's very strange, Kelley. Everything else looks normal. That is. That's weird.
That's ready. Now I just have to figure out what the hell's going on. Here we go.
Hi, how's it going?
How are you doing?
Good,
Good to see you.
coming.
Yeah. Must be exciting.
Yeah, thank you. Your seat of honor is in the middle.
Oh, no.
You're in the
Okay,
rush pot.
all right. Oh, shit.
Get the best seats in the house.
My gosh.
I'm just trying to make my live stream work.
Don't want you to miss a thing.
Richard, do you remember my mom, Joan?
I met you several years ago when Kelley first came. Yeah, we got a backstage tour. Nice to see you again, Joan Jackson. Well, thank you. Actually, we did come by last month. You were there. My brother and I came up. last month. Yeah, that's okay. I totally enjoyed everything all over again.
I've been working mostly 6 to 8 weeks last month, so it must have been that long. I didn't. Yeah. That was really good.
Fabulous again. I enjoyed it again. Yeah.
I'm gonna be right back.
Sure.
This has got a cute little picture of
Love a room with an octopus on the screen.
You know you're in the right place.
Do we know who we're expecting, Kelley?
Your seat's in the middle for one, but no, not exactly. I'm not totally sure because people RSVP, but not necessarily on the.
Richard.
Yeah, Richard's here. Hey,
What's going on?
thanks for making it.
Richard will be here. And
Yeah, he's already here, and Robert
Robert,
needs. I haven't heard from him.
but you're expecting him?
Yeah, but I mean his office is upstairs, so I could,
Right.
if he's here, I could go get him.
Okay.
how we can hear everything. That we just-
Oh, you can hear everything. That's good. Okay.
We're trying to get you committed instead of hearing.
Yes, if I could.
Richard to sit in the hot seat already.
Okay, everybody. I'm still working on this camera thing. I don't know why it's not working, but we're working on
Do you
it.
need to call IT?
Well, it's on my own computer, so it's not really. Yeah. Let's see. That's turn camera off. So now I've turned the camera off.
Really?
Yeah.
And then if you go into your main stuff in your Mac, your camera settings are all A-okay?
Yeah, I think so. Like I'm gonna turn. I don't know what to do.
There's no Skype always. Like actually for me, I had to hit that button.
Yeah,
But-
right.
because this isn't showing up at all. So I think
Yeah.
it's closer. So go into your settings.
Because I'm actually like upload speed of the internet here, which I
Okay,
gotta
some
hope
of it.
not, but.
I've had a Mac for 5 years and I still cannot find everything. Webcam. Fire display button.
That's not it. Disable HD.
Have you Skyped recently from your computer?
Yeah, it's not. It shouldn't be that.
The only thing that makes me think it's just that the green light should be on. So
Yeah, right.
there's another way to test your webcam.
What's keeping the CLAC Center busy these days?
Go to your launch pad.
Yeah, I was gonna, like usually opening up Photo Booth
Yeah.
does it.
No.
Yeah.
Right.
But yeah.
Yeah, it wasn't. Right. So, yeah.
Oh god.
That's right. Yeah.
When in doubt, what Alice Heck always told me to do was Google it.
I did, and it wasn't. Mm-mm.
Oh, yeah. See how they do that? Yeah. That's actually. Yeah. Conservation. So that's cool.
Hey, how are you?
One of the scientists.
Green light's not red light, I'll leave it on.
Jealous.
Ah. Rebooting the Mac will fix no connected camera error. Tried that yet?
Yeah, I don't have 3 minutes to do that.
Well, people can hear you.
Yeah. Well, guys, I'm really sorry about this, but I don't know what's wrong with my camera, and I don't really have time to fix that. So you can listen to me, but you won't see me, and I apologize for that. So again, I'm just going to type it in the chat that audio only, and we'll start in just a couple minutes here. I'm just going to turn the camera off so it doesn't keep trying. I think it has to do with how fast our internet is here.
Yeah, Wi-Fi is
Right,
not
yeah.
the fastest needle in the line.
Not my friend. Hey. Okay, because yeah, it's just like blank. Like there's no, it won't detect the camera. Like my camera light isn't even on. Yeah.
That's what you get.
Yeah. Oh, there's a YouTube icon.
You get people from the cruise ships and things like that.
I don't know. I just did it through YouTube, but okay. Yes, yes. Start Hangout on air. Okay.
Research side is even more than the tourist side.
No, it just says live, and it's like my camera light is still not on, and it's still in the Google Hangout window. Yeah. Yeah, it doesn't even have that. Yeah, I don't. It's not. I don't have time to worry about it, but thank you. I appreciate it. Yeah, thanks. Bye. Robert, you're in the center. Robert. Mush pot, yeah.
You guys give a seating site.
I didn't realize.
Yeah, I didn't either.
But I can do that. Hi.
Face-to-face. Good.
So did you know, so you saw Tracy's?
Yeah. I started thinking about it. I wasn't sure whether, I didn't want to respond directly to Tracy. I thought I'd at least get her thoughts.
Oh, no, I agree. I agree.
Yeah.
On the one hand, it's cool.
Right. Right. It's almost insulting. Yeah.
It's been a while since we were 2 bucks an hour.
Yeah. Well, that's only. All the time, outside. Anyway, I thought it was really good.
Yeah, I think the group's
Are we
assembled.
ready to go?
I
You look like you're ready to go. Okay, now,
did you lose your card already?
put it in my pocket, I swear. I did. It's like I'm gonna put it in my pocket right now so I don't forget.
You're
So
gonna
it was
improvise.
Cal
State Long, so
State Long Beach.
yeah.
And-
a degree, bachelor's in
Yeah, and Santa Cruz, right, okay. Hi, still morning. Good morning, everyone. Thanks for coming. I'm really pleased to introduce Kelley Voss in her master's thesis defense. Kelley has, comes to the lab with an undergraduate degree from Cal State Long Beach. I always get it wrong. Cal State Long Beach. And she's been working for some time now on the influence of behavioral syndromes of Enterochristophleny behavior. And so she brought a lot of enthusiasm to the lab when she came, and she brought a lot of ambition to this project that she's going to present today. So I'm really, really pleased and excited to have her defending. When all goes well today, she will be on to a PhD program at Santa Cruz, where she's already been accepted and ready to move on. So we're really pleased to be able to send her on to there from here. Kelley, it's all yours.
All right, thank you very much. I am so pleased to have you all here today. I know it's summertime and I feel really special to be able to do this. So today we're going to be talking about octopus personalities. And when we're talking about behavioral variability in animal behavior, in the past, for the most part, variations in these behavioral data were ignored just because, oh, it's just noise, it's just error. But in the last 30 years or so, there's been an increasing focus on trying to work out the mechanisms behind that variability, because these things are important. And this variability is examined on both an individual level and on the population and species level, because looking at this variability is, like I said, important both in an ecological sense and an evolutionary sense over the time that a species has evolved. So when I say a personality, I mean a predictable behavior observed in an individual. So when we look at studies of personality, we begin with humans. The study of human personality has really been the basis of all of this research. And like I said, in about the last 30 years is when non-human animals had started to be explored. From human research, we get axes of personality, and the main ones that have been identified in the literature are like a shy, bold behaviors or scale of behavior, sociability, general activity level, aggression, and exploration. And Gosling 2001 is this really large review that looks at the sweeping, so far in 2001, there's been a lot of work since then, but it's the basis of what we know about non-human animal personality research. And See et al. 2004, there's two different 2004 publications that try to establish nomenclature for this field. It's still kind of in the Wild West phase, research-wise, methodologically. So he established the term behavioral syndrome for a population-wide personality pattern. And in invertebrates, there are really, the studies really underrepresent the invertebrate taxa, just because vertebrates are very charismatic and sometimes easier to study. But Crowley, Fisher, and Schuett in 2014 wrote a really nice discussion of why we should be studying personality in invertebrate species as well. And that paper noted that there are 5 papers total that study individual variability in cephalopod species. The Sinemolchanivsky and Sin et al. Studied the dumpling squid and non-octopus species. And there are exactly three out of all of this body of work, studies that have looked at any kind of personality or temperament in octopuses. And that's, the first one was Mather and Anderson in 1993, looking at octopus rubescens. Then Sin et al. 2001 looked at octopus bimaculoides. And Pronk et al., which is the important paper for me, studied octopus tetrachus in 2010. And this personality study has interested me because if you haven't been into our wonderful aquarium lab, this is a picture of our octopuses up in Grant. And our aquarists have noticed, since we've been keeping octopuses, that we'll have more bold octopuses that swim around a lot more, and we'll have shy octopuses that like to hide under the rocks over there. But sometimes we do notice a shift in their personality, that expected behavior from them over long amounts of time. So that was kind of where my project has come from, and brought up some questions about the consistency of octopus behavior over time. So when we're looking at the different ways octopuses could be interacting, we want to look at the different contexts they could find themselves in. So these pictures represent 4 contexts, including a prey context that could interact with a crab, a small conspecific, which is conspecific is another animal of the same species. So maybe a small octopus or a large octopus or a stellar sea lion, which is a common predator. And when we're talking about the differences between these contexts, I've been interested in looking at how the risk inherent in a context may affect their behavior. So up here we have, it makes sense that a feeding interaction for an octopus may be kind of low risk. The crab isn't really going to hurt it as much as octopus is going to hurt the crab. But this stellar sea lion can inflict a lot more damage. That's more of a high-risk situation. When we're talking about conspecifics, octopuses are cannibalistic, and so it's possible for them to eat one another. So there may be some inherent risk for an octopus to encounter another large-sized octopus. But if they're cannibals, they may readily eat this little guy if they come across it too. So it's important to understand that they're different. There's kind of a gradient of risk going on when we're looking at this functional context. And what I mean by functional context is, like I said, like a predator avoidance situation or conspecific interaction situation or a feeding situation. So behaviors kind of fall along axes of variability, and we're going to mostly talk about the shy-bold axis that I brought up earlier. So a shy octopus, for instance, would probably be more likely to retreat from a situation if they're shy, and kind of a middle behavior would be freezing in place and assessing the situation, and a really bold octopus would theoretically approach whatever organism it is encountering. So say we have octopus number 1, and we've observed it several times over time within this context of, say, a sea lion swimming past. And so we've got kind of a mean of its behavior, and that's going to be its personality. So this is octopus 1's personality that kind of is middling between shy and bold. And we observe octopus number 2, it's maybe a little more shy. Octopus 3 is a little bit more. Octopus 4 is extremely shy. The mean behavior is always a retreat. And octopus 5 is the opposite. And when we bring these all together, we can see that when looking at the population-wide distribution of these personalities, we get some clustering around certain parts. So say these 3, maybe not totally bold, but they're kind of on the bold side. So these are kind of the dimensions that I'm trying to examine within one individual, the mean within individuals, and then the mean across all the population, and see how that breaks down into personality types. And so a behavioral syndrome, when we're talking about personality, which is analogous to a behavioral syndrome, is a suite of behaviors that are correlated between either within one context or across different contexts. So say we've got the behaviors in this anti-predator context and in a feeding context when interacting with prey items. Each one of these dots is the correlation for one individual between those contexts. And so the general pattern between that would be considered a behavioral syndrome if they're correlated. And then we see kind of smaller circles, we see clustering, so to speak, and then those could be considered more of a personality type. So the questions that I wanted to ask within my study, first off, I would like to know whether giant Pacific octopus, Centroctopus stillfleeni, demonstrate that kind of behavioral syndrome, that population-wide pattern of personalities over time and across functional contexts. And I also would like to know if they distinguish conspecifics from potential predators or prey, since there's also some inherent risk involved in both. So my first hypothesis was that individual giant Pacific octopuses would demonstrate predictable behaviors, some sort of personality over time and across contexts relative to that perceived risk, whether it's a prey or predator or a conspecific of different sizes. Second, that giant Pacific octopuses would demonstrate the same behaviors toward that small conspecific and prey and large conspecific as to predator, again, that risk. And lastly, I hypothesized that the population-wide giant Pacific octopuses would demonstrate a behavioral syndrome of consistency across days and contexts. So the way I studied this was I used a technique called high definition video playback. It's a fairly new technique within the behavioral studies world, but it's been done just a few times with cephalopods successfully. This is a overhead view of a hypothetical tank. So we've got our octopus here, and it's completely visually isolated except for this television monitor that's pressed against a viewing window, and it can see a high definition image of, say, a crab or all of the things that I showed them. I'll tell you about that. And on top of the television monitor, we have a camera pointed into the tank to capture as much of the field of view as possible to record all those reactions. The video was being played off of a computer so I could manipulate and control the video quickly, being projected onto the TV. And there was always some sort of den or visual barrier so the octopus could self-select to remove itself from the view of the monitor if it was being stressed. And this is kind of the real life view of what that actually looked like. So this is at the Steinhardt Aquarium in San Francisco. This is Beatrice. And she could see the image on the screen. Here's the camera on top of the television. And there's a curtain wrapped around the tank so that the only thing that was changing was what was on the television monitor. So remember that important study from earlier, Pronk 2010 with the octopus tetrachus? This is the first one to successfully do a behavioral study using this high definition video playback technique. And I know that there are a few studies coming up that have not been published yet that are also using this technique other than my own experiment, and that it mainly involves Yuzuru Ikeda's research group in Okinawa. And they're looking at, they've been doing animations of prey to try and do something similar to what I'm doing, but with cuttlefish. So I had 6 test facilities. I tested half of my animals here at APU, but with just wonderful generosity from 5 other aquaria, I got to double my sample size I tested 4 animals at the Alaska Sea Life Center, and I also traveled down to California and worked at the Aquarium of the Pacific in Long Beach, California Academy of Sciences, that's Steinhardt Aquarium, and Aquarium of the Bay in San Francisco, and the Birch Aquarium at Scripps in San Diego. All of the animals were tested in tanks that were familiar to them. So the APU octopuses were acclimated to their tanks for at least 48 hours to give them time to explore and adjust. And the octopuses in the public aquaria were kept in their display tanks. I didn't remove them at all. I just set up within their display area. This is an example of what my videos looked like. I did 3 trials in one day and 3 days of trials. So day 1, trial 1, I showed 180, these are each 180-second clips of a control, which is an empty tank with moving water, and then a conspecific, here it's a large octopus, and then the control again. And then I showed prey items surrounding that control, and it was a crab moving back and forth. And then the next series showed a novel object, which was a bobbing jar. And then they either saw a small conspecific or a large conspecific, never both. So the first 2 days, they either saw the small octopus or the large octopus. So that was trial 1. Trial 2, it's the same idea. And then trial 3, they saw all that video, and then at the end I showed them that clip of a predator, and that was first to be able to compare the reactions to predators across days, but also there was some concern from the aquaria that lion would stress out their octopus, and so we did that just to be sure I wasn't going to traumatize their octopus at the end of the week, so they only saw a short clip. And day 2, similar to day 1, with that the sea lion at the end of the 3rd trial, and either a small or large octopus. But then on day 3, they saw kind of the same order, except they all saw a small conspecific in the morning, a large conspecific on the 2nd trial, and instead of a conspecific, they saw a sea lion on that 3rd trial. And this is a screenshot of what I actually scored, like with the video that was reported that I actually derived my data from. So I counted the number of approaches, the number of head bobs, which is it's a visual ranging behavior, and they kind of do this very distinct up and down, or down and up movement of their head, and the number of retreats. I counted the number of times their body pattern changed, so the number of color changes, the number of texture changes. And I also recorded the amount of time they spent moving around in the tank, like actually traveling, the amount of time they spent stationary, both in a den and outside, just observing, and then the amount of time they spent touching the area where the stimulus was being presented. And when I'm talking about color and texture, we don't have a really good way to quantify that right now. We don't have a body pattern map established for a giant Pacific octopus. So just for the quick rundown, I did a score of between 1 and 5, and 1 was all dark, its whole body was dark. 2, about 75% dark, starting to see some light components on their body. 3 is pretty 50-50, even modeled camouflage pattern. 4 is kind of that light, and then all the way up to 5, which is pretty much a pure white octopus. And similarly with texture, I defined a 1 as completely smooth on the eye and really no pronounced lateral mantle folds, so smooth octopus. 2 had those lateral mantle folds starting to appear. By a 3, they're starting to erect those eye papillae. 4, they have more extension in those eye papillae, but they're also erecting papillae along their body and their mantle. And by the time you get to a 5, they've erected all of their papillae fully, and they're very rugose. So I have an octopus, and I've shown it several functional contexts. And in each functional context, I've collected data on 8 variables. This is a lot of data and not a lot of variability per variable. So what I had to do was a principal component analysis to try and be able to compress those data into a more distinct, like you can tell what the pattern of the variability actually is. So I took all of those data and I subjected them to a principal component analysis, which takes these 8 directions of variability and collapses them into less so you can visualize those patterns again. And along each of these axes of variability, these principal components, each one of those behaviors is accounted for, but it just loads differently. It contributes a different amount to each dimension of variability. So what I did with that information was I took all of those data, all the original data, and multiplied it with their respective principal component loadings. And I used those data in my analyses. And this is it's called a principal component analysis regression method, and it's described in a book called Tabachnik and Fidel, 1996. I ended up testing 6 males and 11 females, ranging in weight between 0.4 kilograms and, which is about this big, to 23.7 kilograms, which is rather large. And the mean was about 5.3 kilograms, which is about 2 to 3 pounds. I analyzed 83.5 hours of video, and that breaks down to about 5 hours of video per animal, for a total of 8,818 hand-entered data records. The duration behaviors were the most commonly exhibited ones. Again, those were the stationary, displacement time, and stimulus touch. And stationary behavior contributed to 79% of the total trial recording behavior. They did a lot of sitting, which is consistent with octopus ecology. And when I'm looking at the important dimensions of variability in this study, it's important to do, it's called a scree test. It's described in Legendre and Swan and Sandilands. And you use, it's called the hypothesis of equal explanatory power. So basically we have all of our principal components along the x-axis and the percent of variance described by each component on the y-axis. And this dashed line here represents that equal explanatory power threshold. So if each one of those components represented an equal amount of variability, they would all line up against this line. And the ones that exceed this line explain more, and those are the ones that we want to address in our study. So principal component 1 represented about 44.4% of variability, and principal component 2 represented 23.7%. So, and we conducted this, or I conducted this with the day 3 principal components, because all of the animals saw equal proportions of all of the stimuli on day 3, as opposed to some saw only the large octopus on day 1 and only the large octopus on day 2, or the small. So I figured that would be the most appropriate comparison. And the way the behavior is loaded on each of the components, this is the same idea of that explanatory, the hypothesis of equal explanatory behavior. But this is equal loadings. So if this behavior loaded equally onto each of the principal components, it would sit along this line. But if it exceeds the line, then that behavior contributes more to that particular principal component. And since we're only looking at the first 2 principal components, we're going to see that these are all the behaviors on the x-axis. We see that the approach, displacement, retreat, and stimulus touch behaviors all load heavily onto principal component 1. And the color changes, texture changes, and number of head bobs load heavily onto principal component 2. So for the rest of this, since these 4 behaviors, the approaches and retreats, all involve really movement and engagement, I labeled these as activity, going back to the axes of personality in the literature. And similarly for principal component 2, since colors and textures and head bobs are all kind of camouflage and predator assessment, risk assessment behaviors, I labeled them as reactivity. And out of 17 octopuses, 11 of them were consistent throughout the trials across days within contexts. 6 varied across days in at least one context, and 5 of those 6 varied in the novel context, and that's kind of a theme throughout all of these data. The novel object elicited a very different set of behaviors from everything else. But none of them were positively correlated across all contexts, so there was some behavioral variation in every single octopus. And we're going to talk about 2 comparable octopuses just for comparison, because I have 17 sets of these tables. We don't want to see that. But we're going to talk about Donald and Ginger. They're both about 0.4 kilograms. They're on the small side, and they're both male. But Donald showed really consistent, really high correlations. These are Pearson correlation tables. Pearson correlation tables between the different functional contexts. And as you can see here, he's pretty much always correlated, except on the activity axis of variability, he behaves completely differently in the novel context. And for reactivity, he's showing different levels of color changes, texture changes for that C line, but everything else is pretty standard. In contrast, Ginger had a lot lower and a lot more variable correlation values. They're all still positive, strong positive correlations for the most part, although you can see here there are some really low correlation values, which means that their behavior on one day couldn't really predict their behavior on another day, or actually in this case, the behavior in one context couldn't predict their behavior in another context. But again, there's this strong negative, well, here's a moderately negative, but negative correlations here when the rest of them are positive, and negative correlations here when the rest of these are positive. So it's interesting that's the same pattern, and that pattern persists in all 17 animals. When we're talking about the population, these are the Pearson correlations between the pairs of days in each context for the population mean, and also the Kruskal-Wallis rank sum value H across all 3 days. And really what I want you to get out of this is that it's all strongly correlated except for those 3 values of activity for novel, their behaviors in the novel context on that activity axis don't predict each other across days. And that H value here is almost significant. So they're almost not different among all 3 days. And this is the weighted state. So I just want to look at the stationary data because that, like I said, accounts for 90 or 79% of all of the behavioral time. And these are still weighted with that PCA regression method because that makes the data normally distributed. So we've got our weighted time score along the x-axis or the y-axis, and all those functional contexts along the x-axis. Day 1 is in circles, and you can see that it's a different spread. Each one of these circles represents the score for each octopus, and so you can see that the ranges and spreads of each of these scores or each of these groups of scores are different in each context. But then when you get to day 2, we could see that those scores weren't really correlated, and this is why the range is so spread out, it's so different. But when we're looking at novel, remember that novel wasn't really correlated across anything else. All those novel scores are up here and down here, and the day 1 scores are here, and the day 2 scores are on opposite. They have opposite pattern of values. And then day 3 looks a lot more like day 1, but kind of the means have shifted. There's more day 3 values lower. And so this low score means that they're moving around less, and high scores, they're moving around more. So those patterns are generally variable between days, but not significantly so. And this is the same idea with those reactivity, with the scores on the reactivity axis. It's the same stationary weighted time scores across contexts. And you'll see that the patterns are also still really spread out, and there isn't really a clear pattern between the 3. Although, yeah, it's kind of spread out. So population-wide, back to these correlation tables, these Pearson correlation tables, just on day 3, just to be comparable between all 17 octopuses, we see that same pattern of the novel being all negative correlations and all the sea lions being negatively correlated as well. So to address the first hypothesis one more time, when we're looking at, this is the personality hypothesis, that individuals will demonstrate this predictable behaviors over time relative to risk. And we did see that personality, that correlation within most contexts across days, although that novel context did show more variability than the rest. And across contexts, those personality types didn't persist across all of the contexts. There's always that difference with the novel and the sea lion. And when comparing those risk situations between a conspecific and prey and a large conspecific and a predator, we saw similar activity and reactivity scores for both the small octopus and the prey item. And with the large octopus and versus the sea lion in that high risk situation, we saw similar activity scores, which were low. They weren't really moving around as much when they saw that high risk stimulus, but their colors were, those color scores were really different. So both the color scores, the actual colors that they were, and the number of times that they changed color and performed those head bobs, that visual ranging behavior, they did less color changing and less bobbing in reaction to that sea lion. So it seems like they weren't trying to engage that sea lion, right? That makes sense. They're employing their camouflage ability to hide and not move and wait for that sea lion to go away. For the third hypothesis, which is the last one we're going to talk about, I'm looking to see whether the population demonstrates correlated behaviors across days and contexts, which is indicative of that behavioral syndrome. And the behaviors are correlated across days, except for, again, that novel behavior and in the activity axis and the sea lion, the reactions to the sea lion and the reactivity axis. And I found that behavioral syndrome was present over time and across contexts. With that, it took a village to get me through this, and I would like to thank my thesis committee first and foremost for hanging in there with me. I would especially love to thank the Environmental Science Department. Thank you to Mandy and Leslie and Professor Shield for bringing me forward and making this possible, because if I wasn't able to present in the summer, I would have missed out on the PhD opportunity. So thank you, thank you, thank you. My thesis was funded with financial support from ATSI processors through my lab, who I would like to thank with a special thanks to Ann Benolkin, my partner in crime, Brittany Ellman, who lent me the tank for recording all of the visual stimuli, and the crew of the 2015 expedition for really heavily helping me collect all of those animals. I would like to thank the Aquarius and administration of the Alaska Sea Life Center, Aquarium of the Pacific, Steinhardt, Aquarium of the Bay, and Birch Aquarium for being very welcoming, very willing to facilitate anything I needed. It was a pleasure to work with everyone. And thank you to Aquatech and Dive Alaska for all of their help in renting the equipment and getting me out into the field. I'd like to thank the crew of Ocean Mysteries with Jeff Corwin. This work was featured on television this spring, which was really exciting. And you can catch it on Hulu anytime, it's on there. And again, thanks to Gordon Scott for his help with trying to collect more octopuses. And I'd like to thank my family and friends, especially Jenny Hoffmeister has contributed a lot to in every step of this. She's a wonderful collaborator that I'm collaborating on a different project, but she's helped me a lot. I'd like to thank Jessica Lee, Danny Estes, and the McFarland family, who all housed me while I was pursuing my data. And last but no means least, I'd like to thank Ryan Friedman and Katie Lyons, who got me into this grad school mess in the first place. And with that, I will take any questions, but remember, be nice, my mom is here. Thank you.
Yes, Leslie.
I've already been nice, so I'm not gonna be nice again.
Right.
Just kidding. So I'm a little confused about some of your statistical analysis, but I'm just curious how you chose the analytical approach you use. Is that based on the other experiments that have used this approach that's in the literature, or?
Yeah, so what they do is, what we do generally in all the literature I've been following is they compare the behavioral scores between contexts using Pearson correlations, and then they use I use Kruskal-Wallis because it's, since my data set is so small, I needed to use a non-parametric test to compare across the days, to compare 3 days across.
So you answered my question because you pulled it from the literature, so that's.
Yes, yeah.
So, so with the principal components analysis, I just want to make sure I'm understanding this correctly. I sort of expected when you started presenting the analysis that you were going to use those principal components in some sort of direct comparison rather than pulling the weightings and then multiplying those by the scores.
Yeah.
And then you compared those back to the PCA scores?
So what I did was I took, okay, that didn't fly, or it did fly, it flew too far. So I took all of the original data, I subjected it to that principal component analysis, and we got those loadings. And by multiplying the loadings or multiplying the original data, all of those variables, counts and scores by their loadings within those principal components, that's how I normalized those data. They were non-normal, and also I needed the non-parametric tests. And so it normalized my data set, and it weighted things because, especially since that stationary variable accounted for such a high amount of the data, it really, those loadings were really very small, like 0.0, 0.2. And so it kind of reweighted everything so I could compare.
So then your comparisons were those weighted scores across the days in context,
Yes.
not comparing them back to the original principal component.
Right.
Okay.
Yeah.
Thank you.
Yes, Brad.
So given that, how would you interpret one of your tables in terms of what's the difference, if you can put it in for us in terms of like what an octopus is doing or not doing, between a 1 and a 0.995 per minute?
Sure. So when we're going, let's go back to, say, yeah, let's do this guy. So if it's like a 0.999, for the most part, say, let's say for, let's compare between small octopus and large octopus, and it's a perfect 1. That means that the octopus was doing, it sat stationary and it changed colors 3 times while it was sitting and watching. It did the same thing for that small octopus, not large octopus. But say, now we're talking about small octopus and this novel, and it's strongly negative. So that would mean that, say that small octopus is sitting most of the time and changed colors 3 times. That novel octopus, maybe it was moving around the entire time. It was really excited about this new object in its field of view, and it changed colors 17 times or changed textures 14 times. So it was, that would be the difference. It's mostly that change in stationary behavior. It's either stationary or not, kind of was a large part of that data set. But is this making sense to you? Are you following like what the?
So it could be a host of different meaning.
Yeah, yes.
Yeah. I guess related, what do you think the implications are for the weighting approach that you took? You know, in terms of your ability to make inference about the way an octopus operates
Right.
in the natural system, and did you, in that context, explore other forms of weighting?
I did not explore other forms of weighting because this was the popular method for cephalopod behavioral research. And the weighting is pretty extreme, but I found that it seems like it was a pretty effective method to me, just because all of the other behaviors would have been completely insignificant if I hadn't weighted it at all. And this seemed to be the best way to weight it and still account for the variability of all of the data set. Does that make sense? So the PCA regression method really considered it, since it reweighted everything so dramatically, just to make all of the other data comparable to those really high stationary scores. Those values are really high compared to the rest of the data set. So
Because that skewed distribution in terms of activity types or whatever you want to call them, but mostly stationary, is that consistent with other octopus and similar studies?
yes, yeah. Sheila and Biss in 2012 found that octopuses really spend most of that vast majority of the time, like 80–90% of their time, just sitting in their den or very localized to their den. They don't really travel around a whole lot. So that it makes ecological sense that the octopuses were mostly stationary, but this did give me a chance to account for both that really large amount of stationary time and also the variability that was occurring in those color changes, texture changes, and actual movements that were occurring.
Can you go back to your loading slide real quick?
Sure.
The one with the bars.
This one?
Yep. So stationary here loads at basically 0 on activity.
Yes.
And it is not one of the, it loads heavier on reactivity.
Yeah.
I imagine because color changes might happen commonly when they're stationary and presented with threat. But in any case, that low loading of stationary into activity essentially means that you're not looking at stationary in that variable.
In that variable,
You're looking
correct.
at these other major ones.
Yeah.
One of the behaviors that you highlighted was, or one of the contexts that you highlighted was that the response to predators sometimes differed from the other responses. Didn't correlate well with the other responses. In the reactivity component, right? So do you have a sense of what kinds of behaviors, what colors and textures are common when they see the predator stimulus that are less common in the other stimuli categories?
I do, and I let me actually pull up a slide that I have at the very end. Well, first off, this is the mean kind of body pattern. These are reactivity kind of mean scores. So, and it actually broke out differently between sizes of the conspecifics that I showed on day 1 and day 2. So I saw kind of higher numbers of head bobs if they'd been conditioned with a small octopus on days 1 and 2 than if they saw a large octopus. And these are just kind of cheeky little graphs about the mean actual color scores. This isn't the count anymore. This is the actual color score I assigned. So this little square in the center, this is the control value. And the triangle represents the score for the sea lion. that black dot is the large conspecific and the white dot is the small conspecific. So I actually found that in those higher risk, those two, the large conspecific and sea lion scores, I found that their rugosity, their skin rugosity, their texture was higher than control. And in both of those higher situations, but for the color was interesting because the texture scores are on this y-axis and the color scores on the x-axis. They were actually darker here. If they had been conditioned with a small octopus, they're conditioned to see really low risk environments more of the time within this experiment. They displayed darker colors toward the small conspecific, or toward, they displayed like darker colors toward both the small conspecific and large conspecific. But if they'd been conditioned with a large conspecific, like a high risk situation, they were more likely to be lighter than the mean, like, or lighter than the control. So the control is really a no risk situation, right? So yeah, I found some interesting differences between the two conditioning types, but they're not that huge, but they are there.
And those are population means?
These are population
for the
means.
two different conditioning
Mhm.
treatments? Thank you.
Yes, lady.
I'm just curious about the videos that you were showing them. Do you have examples of like a predator video and or a small octopus video or whatever?
I didn't put, like, the file would have been huge if I put the actual video in, but these like these are all screenshots of the actual video.
So are the videos, are some of them animated and some of them not animated?
No, they're all live recordings of
Okay.
everything. So that's a real small octopus and a real fairly large octopus. I call it large because it takes up the whole screen. It's kind of magnified a little bit by being projected onto the screen.
And there was motion in all of
Yes,
these.
there was motion in all of them, including that empty tank. The water was moving, so there was kind of light moving through. Yes, Brad?
But just your comments about conditioning, I think are interesting. And the fact that a subset of your observations come from the tanks here at APU, which are in a room behind a closed door,
Mhm.
few interactions on an hourly basis, probably for people, versus a suite of tanks that are display tanks.
Mhm.
Did you compare
I didn't
these
because
as separate groups to look at, some are habituated, or I don't know what the right term probably
Yeah.
is, there's lots of stimulus along the edges of there.
right.
And perhaps really understand that's a glass ball
Yeah.
versus a different group.
Right. I haven't broken that down, but I'm not sure there's a huge difference because I did see some pretty variable octopuses in the APU lab. I also couldn't control because the octopuses in the APU lab, they acclimated for at least 48 hours, but some of them were animals that were already living here when I got here. So Khaleesi had been living here for almost a whole year by the time I actually tested her. So I couldn't really control for the amount of time spent in the tank, and I couldn't control for the number of human faces it was exposed to. So that's something I haven't broken down, but I don't know quite how to account for that in a structured way, just because all their environments are so different.
With these plots, the way, I know there's only 3 days, but you can compare them in general to some form of null model that says, all right, there's just neutral behavior like this, or there's habituation. Like they're now used to being in an environment that's wrapped in a cloak with a camera, and each day the octopus becomes more familiar with this thing that's happening to it.
Right.
If that were happening, what would you expect these to look like?
So say let's use this one for example. I would expect that if they were habituating, this spread would go down, but day 2 the spread kind of got worse. They kind of broke out, and so, but then on day 3 that pattern, it doesn't seem to follow any kind of logical. I would expect these to tighten up sequentially, not have this one be kind of moderate and this one be huge, and then or day 2 be a very widespread and day 3 be the settled, like really compact range. So I don't know that there is a total sense of habituation there, just because the patterns were so different. And
Are there
everyone's,
other null models? So that habituation was just
Yeah,
of another form of a null model that you could compare it to. Are there others that you consider that could explain these observed patterns of relatively large change?
not that I looked at. Within the literature, other studies have found, like the paper that I originally based the methodology off of, as far as the high definition video playback went, they only found, they called it an episodic personality, and that just means they found these behavioral correlations over the course of a day, and then that flipped. So the next day they were exhibiting completely behaviors that couldn't predict what had happened before, and couldn't predict what was going to happen the next round of trials with that species of octopus. So that sort of looks like the same thing here, right? This looks like an episodic personality. It's evidence of a personality, but it's not necessarily over time behavioral syndrome wise is within the group.
If you were to model a null model of what's called random noise or something like that, it says, each animal makes a choice in any given instance, and could be literally based on some host of factors that have nothing to do with the trials.
Right.
What that might look like.
I feel like it'd be an even spread. That would be even for all of if it was random, then they'd all be the same. I would expect the null model would be some spread of, I don't know how far the spread would theoretically be, but I know that it would be the same across context. That score would just be, the scores would all just be.
Kelley, did you read it all in the about responses of octopuses to training, when they're trying to train octopuses to respond to particular stimuli this way or that way, to test, for example, whether they can distinguish A from B? So I know we talked at one point about some of that literature. So one of the reasons I ask is, this pattern that you see, go back to day 1, where you sort of think, well, that looks fairly clear. I mean, some of them are above the line, some of them are below the line fairly clearly. And then day 2 goes to Scatterville. And then day 3, you begin to see possibly some reassertion of some sort of pattern, which you're asking about habituation, for example, as a null model. This pattern that you found is actually very typical of cephalopod training. Okay. They look like they're going to train up really nice initially, and then they get curious, and they just do all kinds of stuff, and all patterns seems to vanish. And then they settle into some sort of possibly adaptive looking pattern that you might expect out of an ecological set of conditioning. And I'm intrigued that you see that sort of curiosity matters as you cross days in this notion of, are they habituating to a stimuli? What does habituation look like in cephalopods? It actually looks a lot like what you've seen. But then you also pick up this signal from the novel object, which is just this curiosity that they don't have, particularly some of these animals in the public aquariums just don't have any exposure to. And there again, you see a very strong response to that, which again hammers on this notion that for some reason cephalopods just have that curiosity knob turned up pretty high.
Right. And also I think part of the reason that this just looks kind of noisy is because it's over a 5-day span. If we're talking about habituation, it might take them more than 5 days to actually get used to this kind of thing. So we might see a better idea of what happens over a longer scale of time.
It kind of looks like they figured out on day 2 that they weren't getting anything to eat.
Yeah.
Based on these exciting pieces of video.
Because that's the other thing. I wasn't training them.
The argument would go the other direction. It only took one cycle to figure out there wasn't any cool that was about to happen.
Nah.
And that, well, to that point, I don't know if it was logistic limitations or whatever, but did you consider the possibility of running much sort of longer term trials than the few days?
Yeah.
Or did you do that with a subset of these?
I couldn't do it with a subset of them because the APU ones, we can only keep really 2 at any comfortable amount of time. And so if I did do it with a subset, it'd be like 4 of the 17 octopuses, and I wasn't able to do that over a longer term.
But interesting trade-off though is that 4 of 17 might be less, but far more consistent than 17 highly variable in terms of their background experience, place of collection,
Right.
all those sorts of things.
Yeah, that was kind of outside the scope of what I was trying to do, but at least this short of it, because all of the other cephalopod tests or studies have really been over maybe 2 weeks. So mine's 1 week, but they're all still really short amounts of time because they are so short-lived. I mean, giant Pacific octopuses are 3 to 5 years instead of the other animals that have been tested that are just 1 to 2. But yeah, I wasn't able to test them for any longer, just logistically. Yes, Nathan. I do.
You made a comment in your introduction that had evolutionary implications.
So it actually has been found in some cephalopod species, the Euprymna tasmanica, the eastern dumpling squid. Yeah. They have found that the more genetically related squid have behaviors that are more similar to each other than ones that are not genetically related. So that's interesting, and I do think that there may be some level of heritability, especially since octopuses don't have overlap of generations, so they have to have some sort of behavioral programming in there somewhere, So I do think that some of this behavior is heritable. Obviously, there's going to be some environmental factors to behavioral variability, but I think there's some programming in there, yes.
Kelley, what would be your thoughts on other senses, incorporating into this? You've got factory and tactile and
the chemosensory stuff.
Range from leave me alone, I'm not here, to volcanic, when you have other things going on with them.
Yeah. I'm sure that those other sensory inputs do have an effect. I could only, the reason that I chose the high definition video playback is that it was repeatable and consistent and easily manipulated and repeatable. So I could take it to wherever I needed to and make the exact same thing happen too. So yeah, I've seen other, I've only seen that I can recall stress tests, seeing how stressed the scent of a predator makes a cuttlefish. And they tried a bunch of different scented waters from different tanks in a flume. And it turns out, because they're measuring respiration rate as a analog of stress, and actually they were more stressed out about finding food. When they smelled food, their respiration rate got much higher than when they found a predator. So it's definitely a factor, definitely something to consider, but not anything I could control. Yes, Sarah.
I assume that weight correlates to age of the individual.
Generally, but octopuses, their weight fluctuates with feeding rate. So if an octopus, like in our lab, we keep octopuses, we feed them regularly. They're fed, but they're not fed at a exponential growth curve. They kind of controls their weight, which controls their size, just because our tank is not that large. But if you feed an octopus, one of the octopuses at the Sea Life Center, when I tested him, he was this big, and then when I came back the next year, he was this big. So it really just depends on how much, what their food intake is.
I ask only because I was curious if you noticed any correlation or observed any correlation between the degree of variability on an individual in a reactive context and their likely age and experiences.
Yeah, they're in other cephalopod species, they've found differences in temperament, which is kind of same thing as personality, over like ontological stages, like over different age ranges. But like the two that we compared, the Donald and Ginger, they're basically the same age, they're the same size, they're young of the year, giant Pacific octopuses. So, and they showed two different patterns of variability. So it's possible that it contributes, but I just don't know how much. Anything else, Peanut Gallery?
Any questions? All right, thank you.
Thank you very much for coming.